Good evening, ladies and gentlemen. Welcome to this Convergence Science Network presentation. My name's Andrew Loney. I'm the Director of the Victorian Life Sciences Computation Initiative and the EMBL Australia Bioinformatics Resource. And on behalf of the network's partners, it's my great pleasure to welcome you here this evening. Melbourne's position as Australia's preeminent medical research centre was again reaffirmed with the results of the NHMRC, that is National Health and Medical Research Council's 2015 project grants funding announced recently. Of the total funds available of almost $420 million, Victorian institutions received around 45% of the total. While this is clearly a very strong result, only 14% of the total applications across Australia were funded, which is the lowest result ever, and underlies the challenges ahead for medical and life sciences researchers in Australia. The research community, and we hope the broader community, supports growing investment in continuing to develop our research capabilities, which is so critical to addressing many of the health issues that confront our society every day. It's no surprise that with the latest NHMRC funding results, Victoria also dominates the nation in the generation of biomedical research data. My organisation, the Victorian Life Sciences Computation Initiative, provides strategic support services to enable Victorian life sciences researchers to transform their research through access to the VLSCI supercomputing facility. So we are well aware of the vast amounts of data that biological-based research is capable of generating. With a significant volume of life sciences research undertaken in Europe compared with Australia, Europe has some 500,000 life science researchers, one can only begin to imagine the issues associated with data generation, storage and sharing across many more institutions. Fortunately, our speaker this evening is tasked with developing a research infrastructure that consolidates national life science data centres, services and core bioinformatics resources into a coordinated infrastructure which is used to translate life sciences research to medicine and the environment and the bioindustries. This initiative is known as ELIXIR. Dr. Niklas Blomberg is the founding director of ELIXIR, the Pan-European Infrastructure for Biological Information, since February 2013. Please join me now in welcoming Dr. Niklas Blomberg to deliver his talk, ELIXIR, How Europe is Dealing with the Exploding Demand for Life Sciences Data Across the Public and Private Sectors.
Thank you very much for that kind introduction, and thank you very much for inviting me here. It's a true pleasure to be here in Melbourne and talk about ELIXIR. Geographically, my body is still in Europe, so maybe a few of you can put your phones back on so you wake me up occasionally. So what I'd like to do tonight is try to explain how we are beginning to connect national investments and national efforts to deal with big data in biology, and also try to explain why this is important. I understand it's quite a mixed audience, so some of this will be a little bit technical, but I'll try to motivate and explain also for a broader audience why I think this is important, both for the research community, but actually the society at large. But actually big data in biology isn't very new. So there has been infrastructures to deal with big data for many, many years. And this is actually one of the first ones in Europe. This is the Botanical Garden in Padua. So this was funded in the early 16th century. So about 10, 15 years after a real invention brought back the first sets of big data for biology from the New World. From Columbus brought back 10 unfortunate Indians, a few parrots, and as much gold he could load on his ship. But really what happened in the early 16th century is that a lot of new biological material hit Europe. And they really tried to understand, how do we deal with this in an infrastructure sense? Because you need to use it for research, but you also need to use it for teaching. And Botanical Gardens, interestingly, was founded at many medical schools and universities in the early 16th century just for that purpose. And so they were used there for research, to grow the plants. They were used for teaching, to make sure that the students understood the medical plants. But they were also actually part of an open data network, because there was a lot of exchange of seeds between the botanical gardens. Interestingly, they were also secure. So this one had a big wall around it, and it had quite severe sanctions for unauthorized data use and data leakage. So they were really quite concerned about the IP aspects of that data. And of course, if you managed to cure a king or a prince in Italy, your fortune was there. You would live happily long thereafter. So the reason why I think this is a really good metaphor for what is happening now in biology with new assay methods essentially unleashing a flood of data is that, I mean, botanical gardens isn't a random collection of plants. There is a lot of systematic organization of the plant material, and there's a lot of very careful curation and annotation of the plants, and that is what makes them valuable. That is what makes them into research infrastructure. And what I try to argue tonight is that we actually need to deal with modern biological data coming out of, say, the genome sequences in much the same way as people dealt with biological sequences 400 years ago. The other part of life science is that data is actually very heterogeneous, and so we all knew that to understand the body you need to take a lot of different elements into account with a few interesting biopsies. But I think this is again one of the real challenges in biology, is that we need to deal with very, very heterogeneous data, and we need to bring that together actually to understand the complete picture of an organism. And I think this raises a lot of challenges for how we deal with data standards, but also how we deal with data exchange between different laboratories. The third challenge that we face in biology is actually the geographic distribution. So this is a map of next generation sequencing or modern sequencing machines in the world. It's from a resource called omicsmaps.com. But actually what you can see just by a quick glance is the ability to produce very large data sets in biology isn't occurring at the one single site. It doesn't occur at the big telescope or at the large accelerator. It occurs essentially in every university, at every research institute across Europe or across the world. And so the challenge we are facing is a very different data management challenge to many of the other disciplines in science, because people in, say, Melbourne do need to get access to reference data from essentially all of these other laboratories across the world. If you have sequenced a set of breast cancer patients, you would quite like to compare their tumors to breast cancer patients from Finland or from Spain or from the west coast of the US. And so we are faced with this very significant logistical problem of bringing together a lot of reference material in a consistent way and then distribute it back out into a very large and spread out network. So this is what we are trying to achieve with ELIXIR, and this is why we have chosen a very distributed strategy, which essentially is about connecting national centers. We talked about the botanical gardens and the maintenance, and this is of course also true for scientific data. So this is a graph from a paper a few years ago where they actually looked at how likely is it that you will retrieve old data sets, things that have been published and deposited somewhere, and the author has stated that the data is available, and then they just went back and tried to find it. And actually, it turns out that scientific data on average disappear at the rate of 17% per year. And so if you go back 15, 20 years, there is really not a lot of data preserved. And I think the data which is preserved is the data that has been submitted into some of these core biological archives. So in biology, there has been a tradition for many years to put genome sequences, protein structures into well-curated archives, and then they have been stored and made accessible for the community. And this data is, of course, still easily available. And today, the rates are really going up. So if you look at, say, GenBank or the ENA, they have a deposition every 6 minutes of a genome data set. If you look at them, and so this becomes a bit technical, but core biomolecular archives today, if you bring them together, is more than 10 petabytes of data. So this is actually more than most national supercomputing centers have at their active disks. So for instance, Denmark just invested in a large new supercomputer for all the life science researchers in Denmark. They spin about 4 petabytes of disk at that. So even if they wanted, they can't get access to all the reference data in Denmark. We need to find a different model. So why would you want to actually make data available back to the scientific community? And I think those of you who follow the open data debate knows that there is a lot of discussion around the reproducibility of science and the need to be able to verify data. But actually, I think the core argument for making data available to the scientific community is what we call secondary data research use, i.e. scientists are looking at old data and they make use of it. And in biology, this has happened again and again and again, and we know that this is valuable. The problem is to capture this value, and actually now as the volumes and the heterogeneity of the data go up, try to manage the annotation, the user bases, and actually the standards, the citation of this data, because it's requiring suddenly a very, very different scale of investment that we've had before. And these are the sort of realizations that drove a fundamental change in the approach in Europe, and that's what ELIXIR is about. And so if we look at biological data, I think we can summarize that in a number of challenges. So one is, how do we actually deal with this scalable annotation and curation? How do we get people across the world to actually annotate their data such that they can be reused by other parts of the research community? How do we actually take these large data sets and make them accessible to researchers in a very large and geographically spread research community? And how do we secure that this can happen over long term? Because it's no good to save all this data if it just disappears a Tuesday in March. People need to be able to trust these resources, otherwise they cannot plan research programs on them. A lot of this, and I think this is interesting now with biomedical research, is making increasingly use of human material. And so in essence, humans are becoming a first-class model organism for science, right? We have our genome sequenced, but we also walk into these very large phenotyping centers which store all the phenotypic data. They're called hospitals. And almost everyone is walking in. And of course, what is happening now in biomedical research is that we increasingly start to make use of data from humans as to understand diseases, which of course makes total sense. But it raises a lot of issues around privacy and ethical concerns. And I'll come back to how we are trying to deal with this. And then perhaps an even larger challenge is the interoperability. How do we get the data to play together? How do we get different organizations to play together? And how do we do this over different countries? Because science is an international endeavor. So how do we do this over different countries and make sure that the different funding structures, the different research programs, and the different priorities of a number of different countries can play together to set up such an infrastructure? So maybe we shouldn't bother, but I think this is a really good picture, and I think it just argues why this is necessary to do some coordination. So if you look at electricity in many countries, there isn't enough strong governance, support, and long-term planning for coordinated infrastructure. And the solution is, of course, that everyone builds their own little generator. And I think this, in biology, this has worked very well. This method has served the community very, very well for a number of years. But it doesn't scale very well. Now suddenly we can't have 6,000 generators. We need to find a different way of dealing with the infrastructure. We need to move up to a large coordinated infrastructure. And I think all of us know how to connect a battery to a light bulb, but that doesn't mean that we know how to build and wire up a house, let alone how to build a grid for a country. And I think this is where biology is facing an inflection point, and this is what we are trying to address. So what happened in Europe is a number of years ago, countries came together and said, well, we need to build a coordinated effort to deal with biological data. This is called ELIXIR, but fundamentally ELIXIR is about connecting national centers and resource providers up in a coherent and distributed network, and connect that with centers like EMBL EBI that host most of the large reference data sets. So we are funding collectively international organizations like EMBL EBI to host our reference data, and ELIXIR is then about connecting these reference data sets with all the universities and institutes out in the countries. And so if we do that well, we should have something which is stable and robust. And there is actually quite a few of these institutes. So if you look in Europe, there is about, right now in ELIXIR, we have about 130 resource providers and institutes involved in the infrastructure. So that is a large number of stakeholders. They are spread over currently 15 countries, plus that we have a set of countries that are on the verge of joining. We're also talking to a number of additional countries that are sort of on their way into the infrastructure. But essentially, I think most countries in Europe, and this is an initiative that was initially funded by the European Union, hence the focus, are realizing that we need to find a different model to deal with data. I think the other part of this, and I think this is really interesting to note, is that if you look at this part, this is about cooperation in science. This is about getting scientific institutes, the stakeholders in science, to work together. But actually, we are an intergovernmental organization, so we have the long-term support of ministries and governments. So when we develop standards and services, they are likely to stick around for a long time, which is a prerequisite for the institutes to build this into their infrastructure and the local processes. So ELIXIR is based on the concept of national nodes. I think what is important to realize is that these nodes are then funded nationally, and they really are part of the national research environment and the research infrastructure. And they do deliver support to national researchers, and actually a number of these nodes have quite significant consulting roles. So they have between 15 and in the case of SIB, I think about 80 people who are actually tasked with providing guidance, support, and help desks for people for researchers in the country. So it is really a support organization for biology. But it is also a chance of displaying and promoting national flagship projects. So if we look at ELIXIR Sweden, it's represented through something called BILS, which is the Swedish national infrastructure. It's a national research network. It's been around for many years. It predates ELIXIR, and actually many of these, the national nodes predates ELIXIR. But actually in the context of ELIXIR, they're focusing on the national flagship project, which is called the Human Protein Atlas. And so what we are trying to do within ELIXIR is to make sure that this big national resource is connected well and play with other protein resources in Europe. So in the Human Protein Atlas, and this has been funded for 10 years, what they are trying to do is, or what they actually have done now, is to generate antibodies against every protein in the human genome. So they have about 20,000 antibodies. They have taken these antibodies and stained an array of different tissues and also different cell types, and taken photographs of these. Each photograph has been manually annotated by a team of pathologists, and so it's a very high quality resource that they've then made available on the internet. So anyone who's interested in this, and it's actually really quite fun to go there, you just look at proteinatlas.org and you can start to look how different proteins are distributed in the different tissues in the human body. So that's a hugely valuable resource as a reference for protein research and for biomedical research. This has been going on for a long time, and actually what we realized within ELIXIR is that Human Protein Atlas didn't play along very well with the other two major protein resources, PRIDE, which stores a lot of the mass spectrometry data that captures protein distributions in the body, and UniProt, which is the reference database. And the reason why they didn't play along is not because they don't like each other or because people are evil or stupid, but all are long-standing big science projects that have been running over many years with many people. And so you don't change them overnight. And it turned out that early on in the life cycle, each of them had made different choices when it came down to how do you identify proteins, how do you identify cells, and how do I identify tissues. And so what we have done within ELIXIR, we've started to connect them up, and actually the Human Protein Atlas now employs 2 extra people just to make sure that they convert their way of identifying protein, cells, tissues, and other elements into the same way as UniProt and PRIDE. And conversely, PRIDE and UniProt now reference the Human Protein Atlas. So this is interoperability in practice. It makes it easier for researchers to navigate the protein universe, but these things don't happen without effort. They take coordination, they take agreement on standards, because once you start to do the manual mapping between the resources, you really only want to do that once, and then keep on doing that consistently over a long time. Because these resources have been existing for 10, or in case of UniProt, maybe 20 years, and they will be running for another 10, 15, 20 years. And so they do require long-term planning. These are infrastructure projects. You don't change them overnight. And so you need that consistency and that stability for these things to happen. And so within ELIXIR, we'd like to make this much more systematic. This was one of our first projects. And so we do that based on essentially 5 platforms. We want to make sure that we connect and sustain the data resources in Europe. We want to make sure that these are associated with compute and data networking and cloud resources that make it possible to work with the data. We want to make sure that we have standards and that we have interoperability. And there is loads of standards. So actually the problem isn't finding the standards, it's getting them used and having mappings and services that people trust over long time periods, so they bed them into institutional metadata templates and things like that. We want to make sure that all of this can be used by an ecosystem of interesting and valuable tools, and we need to train. And I think most of you know that there is a huge skills gap, and this is also true in Europe. We're investing in quite a big training program to make sure that we actually provide training for the next generation of scientists, but actually also for the current generation of scientists in using all this infrastructure. I thought I would show a few examples. So if we look at data, because data is really the foundation of this, we are doing some fairly sort of infrastructural things about looking at frameworks for life cycle management and making sure that we have high quality curated resources. But actually a lot of this comes down to how do you get the data available in the first place. So I thought I would just share a few reflections on open access that came from the Swedish National Genome Center. So they just asked their users, are you aware of the guidelines for open access? And two-thirds said no. And that is despite that the Swedish Research Council, which is funding most of this research, do have very clear guidelines on open access research. And if you're not funded by the Swedish Research Council, or more commonly you're funded both by the Research Council and the European Commission, the European Commission also have guidelines on open data management within Horizon 2020. And so I think there is a significant knowledge and skills gap, and actually most scientists don't really understand how to make the data available. Although if you accept that you need to make the data available as open access as often part as the publication procedure, you can ask the question, would you like support? And then overwhelmingly, if you ask people if they want a free beer, they more often than not say yes. But actually, I think what it says is that a lot of people actually want help to submit to public repositories. Even more important, they need and want support to do the metadata annotation, to make sure that what comes out of the machines and their labs is annotated in a way that it's reusable by other scientists. And this is, again, a big skills gap and a big need. And so this is part of what we are now trying to support each of the ELIXIR nodes is to put in place through what we call a data nodes network, both the skills and the tools to help the communities to do the metadata annotation. And so one example of this is, so this is about then simplified data submission access and make sure that we have policies and guidance for the researchers so we can sort of percolate the data up from the bottom. One example of this is, again, in proteomics, so working with PRIDE, which I described before. So PRIDE is capturing essentially mass spectra from proteomics experiments. It needs quite a lot of both data and metadata in a submission. It's quite complex to bring together the data package to submit. But actually, if you want to publish in the major journal, you do need to make your data available in a repository. And so there's quite a few scientists who want that. If you look at Sweden, again, they have made available a national infrastructure, because in Sweden, as in most countries, you do need to store research data for at least 10 years after publication. And so the major mass spec labs in Sweden, they came together and built an environment which they called Proteus, which essentially uploaded data from all of their labs and put it in the national long-term archive, which is called SWISTOR. So that would be really convenient if you already have this process right, and from your lab, data goes into the National Long-Term Archive, and it sits there. It would be great if that just could push it out to PRIDE. And so with the push of a button, you would have made that publicly available and fulfilled all the criteria. And so again, we ran a project. We were very ambitious in the beginning. We settled down eventually, but this is actually what the end results look like. So when you do set up and submit into the Proteome Exchange, you can actually just upload the data from the National Archive. And then when you search for your data set in the Open Data Archives, it's then annotated up as coming from Sweden. So you can pull together the Swedish Research Council, and the infrastructure will also be able to generate statistics based on how often these data sets are used and so on, which helps them with the long-term sustainability and business cases. But I think that this is, again, a good example of how we want to build up gradually these networks for data sharing by actually leveraging the resources that in many cases are already there, but it's just a little bit complicated to make them available to others. And so if you look at the broader problem then of data roaming, it is really this access of large and growing reference data sets, and it's the access of secure data sets that is quite challenging. And so one of the things that we've done for the human is to build on something called beacons. So beacons is just, as a beacon would be, an advertising service for data. So within human genomics, you can, through a beacon, you can ask the question, do you have, or do I have a variant, an allele in my cohort of data that is interesting to your research? And so if the answer is no, you don't need to bother with my cohort anymore, right? There is nothing there for you. If the answer is yes, then you can embark on this rather more laborious journey of sharing human data, which is only consented for research use and needs to be handled in secure environments. And so that is the whole idea of Beacon through the Global Alliance for Genomics and Health, which is essentially a bottom-up interest group. There has been a development of standards, but also security policies for managing and dealing with this data. And so what we are trying to do in ELIXIR is to build on these standards and then embed them into a number of national nodes. And so we have begun with 5 countries: Spain, France, Netherlands, UK, Sweden and Finland, to set up beacons. And actually where we are right now is that we have a working technical implementation of how you do this. And what remains to be done is all the governance and the ethics and the security framework around sharing human data. And so this is again, I think, one of the major challenges for life science right now, is to manage the processes of collaborative biomedical research rather than the technical part of collaborative biomedical research. The way it looks, I think, is a good example for this. So essentially, you can share data in this framework at 3 levels. Green means that it's published on the internet. Anyone who wants can go and have a look at this data set and ask for variants. Now, of course, this isn't very secure in the sense that if you ask for a rare variant and you know there is only a very small number of people who have that variant, it's quite easy to understand who actually is part of a research cohort. And this is called DNA-based subject identification. It's a known security fault in any of these sharing initiatives. If you do things openly, then it will be quite easily to leak. So for a few data sets, and it is really quite a few data sets, this is not a problem. The study participants, and they're mostly healthy volunteers, they have consented to have their data available on the internet for free. So this is, for instance, the 1,000 genomes in Europe or the P3G project in the US. Most projects So the vast majority of projects, the people, the study participants have not been consented for this. You can't share data like that. And so what you have is that you actually need to apply for access, and then following a review by a data access committee at the institute, you will get access to the data set. That's quite complex, but actually what we're looking at within ELIXIR is an intermediate category where a number of registered users, i.e. scientists at reputable institutions that sign up to a code of conduct and a usage policy, will be able to ask these type of questions in a somewhat simplified ethical framework. And so I think there are ways of managing the ethical and security aspects within data sharing. So what we are doing is essentially to roll this out now and to use that to test some of the technical solutions, but actually fundamentally this is also about helping countries. In most cases these are national genomics cohorts, to help them to set up these governance processes that lead them, that enable data sharing. And that is not easy. For Netherlands, actually the submission to EGA took 4.5 months. Part of this is annotation, and a large part of that is to understand the governance processes for downstream data sharing. Just moving on. So this is for human, but actually, I mean, biology is much more than humans. And you would like to do the same in the agricultural sciences. So we are right now looking at plant sciences. And I think plants are really important. So, I mean, the climate is changing. And it's a good slide today when the Paris meeting is opening. But I think, at least for us in this audience, I assume that most of us take that for a fact. And so actually managing agricultural research for drought resistance is actually quite high on the research agenda, and something which is really important. A number of countries are doing large-scale genomic sequencing projects for important crop species or agriculturally important species. A lot of these are also phenotyped, i.e. you try to understand the response of the plants to the different environments in quite some detail. And so there are a number of high-value data sets. These are actually quite difficult to share, partly because there has been ill-defined standards of annotation, or there hasn't been really any community-defined standards of annotation, and partly because it is actually the phenotypic data can be quite sensitive or high value, so it may be difficult to share it openly on the internet. So what we are trying to do within ELIXIR, and we've now enrolled 6 nodes on this, that where each of them have picked one of their model species, or one of the important species where they are doing full genome sequencing through a national program. And we want to publish the genome together with phenotypic data sets from these national activities. For the genomic part, so the DNA, this is relatively straightforward. You can just submit it to one of the big archives. Annotation of the genomes is often very sketchy. So even the big consortia like the wheat and the tomato, I think have there is a lot of work still remains on the annotation. For other species, and we have cork, oak, and spruce, and a number of different plant species that have high industrial value, the genome annotation lags behind. And so we are looking at agreement on standards, both minimal information standards, but also the choice of ontologies, so encoding methods and data formats for these phenotypes. This part of the work is right now ongoing. Once this is starts to become a bit clear, we are looking to implement a common API for data sharing. So this will be hopefully something like the beacons, but for plant genomes. But it's going to take some time, but we hope that at least this part will be ready sometime during next year. So again, it's focusing on data discovery and retrieval, but in this case in the agricultural sector. And we're starting to build up a user community around the plant sector for this. So the third problem then that I talked about in the beginning is interoperability. So how do you actually connect all of these things together? And I like to start with this quote, which comes from actually one of the members of our scientific advisory board, Jeroen Wojcik. He runs a small company that does biomarker and translational data management for pharmaceutical industry. And he just said, well, about 40% of the effort on a typical project is actually on data integration. It's not about the value add. It's not about understanding the data. is not about understanding the customer's need. It is really only about getting the data aligned so that we can start to do the science. And the reason why this takes a lot of time is that it's actually quite complex, and most of our data sets have been encoded in many different ways. And I think we saw the example from the Human Protein Atlas. But the other reason why this is complex is because that you ask a lot of very different questions in biology. You ask things like, what compounds have been described in a document? And so then you need to describe compounds, you need to describe documents. What targets does a compound bind with an affinity greater than, say, 10 micromolar? Then you need to encode affinity and binding data. If you try to associate diseases with drugs, well, then you also need to capture clinical trials in different studies, and you need to capture the mode of action. And so there are very many different concepts that you need to encode consistently. And this is challenging. So this is one example. This is for identifiers. So for most bioinformaticians, 9606 is a well-known number. It's the encoding for humans in the NCBI species taxonomy. But actually, if you go to the bold taxonomy, it's encoding a fungi called Bombucilla sedodrum, but it can actually, in the green taxonomy, the same number will encode a plant. It is a chemical if you go to the PubChem database. It is a gene if you go to the Global Gene Nomenclature Consortium. And it is actually quite a few documents if you start to look. And so you can also look up a paper with this accession number in PubChem. And so actually, how do you identify things is often quite ambiguous. The other thing is that we actually quite like to call things different names. And so this is paracetamol, as it's called in Europe. I don't know what it's called in Australia, acetaminophen. But actually, if you build a dictionary of all known names for paracetamol, so this is the chemical structure, you find about 1,100 different synonyms for this compound. And so it's not only that the same name or number can correspond to different things. One thing will more often than not have a lot of different names in different databases. And so that's, of course, a challenge. And so we are launching an activity on managing identifiers consistently within the ELIXIR infrastructure. And so what are the different types of services? How do we manage the life cycle? If something disappears, can you recycle it and then introduce ambiguity, or is that name forever gone? And how do you actually, if it's forever gone, how do you manage forever gone? How do you fit them together? How do you map between different identifiers? And believe it or not, but this is actually quite an active field with a lot of competition. So there are many organizations that would like to own the total problem of the world's identifiers. And so actually we are working right now to try to understand how do you navigate this space in a consistent way and try to provide guidelines for the poor scientists out in the country who just want to say paracetamol in a paper or in a database and be done with it. And this matters, and so this is work we're doing with the rare disease community, where you try to map up some of these data resources with samples held in biobanks and with clinical registries and patient records, because they would be asking things like, I'm looking for skin biopsies of a patient with a rare disease that has a mutation in a specific gene, but only within 2 age groups, or compared to age groups, and I would like to have enough materials so I can do immunohistochemical studies. So this is not an uncommon question from a rare disease researcher, but actually how do you go about answering all of this, because you need to map between a number of different resources. And so we are trying to pull together a set of services from different countries that support this, but it's not easy. So I just showed one example. How do you encode, in this case, marriage in a clinical registry? And so these are 3 different registries. For education level, this one has 7 levels, this has 9, this has 7, but actually you can map them to a consistent ontology. If you look at marriage, you could describe someone as never married, married now, separated, divorced. They could also be in a partnership, they could be widowed, of course, and depending on how you choose to encode your data, this will be more or less difficult to map together. Age and date of birth should be relatively easy to capture, you would think. But actually, in different registers and patient journals, you would say, well, age at the baseline when the study started, that's what you've captured. In others, you will say exact age at visit in days. And so again, you will need to do a careful mapping. Sex is another one. It should be relatively easy. Are you male or female? But then how do you deal with mutations such as XXY or XYY? And how do you do that consistently? And if you look in patient registries, almost every patient registry have found a different solution to that problem. And because it's very uncommon, so more often than not, it's just a text note. And so I think we do face massive challenges in how do you encode data consistently. And the approach that we have taken is actually to go about this through what we call bring your own data workshops. That should be relatively familiar in Australia. But essentially, researchers with different problems come together with data annotation experts, and through a 2-day workshop, we try to bring together a number of different resources. And in the rare disease field, we now run 2 of these workshops in 2015, and this has been very successful. So we're now bedding it in as a program together with one of the major rare disease funded research projects in Europe. So all of this comes back to data management, and so I think you can do quite a bit. We did work last year where we looked at the principles and the guidance for data management across a number of different research infrastructures. So these are people doing crystallography, doing microscopy, the biobanking people, the clinical trials people, and we just mapped out and we actually agreed on a set of 8 principles going forward for how we should drive identifier and data management within the infrastructures. So that's great, right? You come together, you have a workshop, you go for a nice dinner, you go to the bar and you agree. It's not very difficult. And then what happens then? and so actually what we've set up, and I think this was really interesting, you actually work with and develop a course material and training, but you don't train the scientists because most of this happens in very large research programs. So all of them have research managers or project managers that are responsible for maintaining the whole research program. So we actually developed a curriculum and a course for project managers of large research programs for doing and supporting long-term data management. And I think this again is a way going forward. Biology is moving into the field of big science. You are often running this in quite large endeavors, and so it really is about targeting the right people in the organization with that training. So all of this is really quite nice, but then the question is how do you actually sustain this going forward across a number of countries? And so what we have tried to do is to build a strong and organized scientific base in each country, and this is what we call the ELIXIR nodes, to identify key scientific collaborations and the champions that really bring together a critical mass of key scientists within a country, and then try to work with these scientists and the funding bodies to build a long-term governance model for data management that works within the national funding framework. And because of that, you can actually build up strong relationship and trust so they understand what you're trying to do and make sure that the program complement and not compete with the national research priorities. And I think this is really important for this to bed in and work long-term. And so for this to work as a coordinated effort, I think one fundamental aspect of this is embracing diversity. So Europe, and you can think about it, in Europe you will have the coffee axis of very black and coffee in small cups in Southern Europe, going up to rather bland coffee in large cups in Northern Europe. You will have a bread axis going from dark, dense bread in Eastern Europe into very light pre-sliced bread in the more western fringes of Europe, and I mean it's a very diverse continent, and we will have to work with that. We will have to work with all the breads, all the cheeses, all the coffee, but also the national systems, the regulation, and especially for human data, there will be very different legal and regulatory requirements in the different countries. I think the other part is really important to understand that it's a federation, it's a partnership of equals. It's not about a small institute feeding a national infrastructure, feeding some sort of glorified large European institute. This really needs to bring value locally. The data management and the standards and the compute approaches that we're putting forward needs to work with every scientist everywhere. And this is not really important because participants are funded locally. And so this comes down to training, and so we have set up a European-wide training network with where actually representatives from the national training centers are meeting face-to-face every 6 months and a TC every month. And what they are looking essentially is to share training materials, share courses, but actually also we recently looked at e-learning. So it's no good if we in 17 member states develop 17 different e-learning platforms for bioinformatics. That would make it very, very difficult to roll out courses. And so there is actually also standards to be done in this field. What I wanted to end with is really, so I've talked a lot about science and the academic science. I think what I started with saying that this is also part of what needs to be done for industry. And so I'd like to end with a little bit of how we work to make sure that this is bedding into the larger sort of societal framework, and how we can make partnership with industry to make sure that that they can use it. And we've looked at a large European public-private partnership, which is a funding program for pharma and academic researchers to come together through different projects. We're looking at things like semantic data integration, but actually one of the things that we've been looking at is how do we help these projects, which are part funded by industry, to actually submit and maintain their data long-term and make that, release that into the academic research community, which is technically easy, and actually industry has come around, and most of the pharma companies in Europe actually do want to share their data, and they would like to make it available for the research community. But again, there is a lot of IP, legal and regulatory requirements that we need to work through. And so this is, we've started this in a pilot project that I hope will be very informative going forward. And the reason for that is, of course, that industry well know that data, public open data, is intensively reused. So the example I'm showing here is from the protein structure field. So this is images, for those of you who are not biologists, these are images of proteins, the smallest parts of our bodies, binding into different drugs. And actually, most of my background before I did this is in pharma, so these are all projects that I've been working on in a pharmaceutical context. So this so this is a kinase, this is a serine protease, and this is a nuclear hormone receptors for those who recognize protein structures. But actually what is interesting is that the protein structure community has come together for many years and deposited all their data, made that available to the broader research community. So the Protein Data Bank, the PDB, was founded in 1972. And data has been available since 1972. So I think it's a really good example of what happens when you make data available over a long time frame. And actually what happens is that in 1986, so this is like 15 years after the database was started, then there was enough data that Janet Thornton and others realized that you can start to use statistics to understand how proteins are built. And so this essentially seeded in the mid-80s the field of structural bioinformatics, which turned out to be a whole new research field. And I mean, there's thousands of papers published in structural bioinformatics every year. And so that would not have happened if the data wouldn't have been there. And there is no way you can plan for this. I mean, you can't say, well, 15 years down the line, I'm sure someone will come up with a really smart idea for beta turns. It doesn't work like that. It's just there. And I think what is even more interesting now, the volume of structures are so that a lot of this is automated. So this is from an automated model assessment environment, but this is essentially a community competition or benchmarking where a number of people working in structural bioinformatics have set up an agreement. So every Thursday night, all the new structures from PDB is downloaded to a server, and they try to predict how they would look like. And then on Friday morning, this is released to the public, and so they can score back and see how well they did. And they do this every week, and they've done that for every week for 6 or 7 years now. And so, of course, this really builds trust, but it also drives the science forward in this way that a closed database wouldn't do. There is a lot of value here. But this is one example. What happened about 6-7 years after this one is that Hans-Joachim Böhm, who was then at BASF in Ludwigshafen, so a German pharmaceutical company, he realized that there was enough of these small molecule protein structures with binding data available in PDB that you could actually start to derive scoring functions, i.e. Functions that describe the affinity, say, of an inhibitor to a serine protease in the body. And so he developed then in the early 90s the first scoring function and then implemented that in a program that he called LUDI, for Let Us Design Inhibitors. And this essentially started the whole field of molecular modeling and docking. And so, as I said, my background is in pharma, so for 10 years, every day at work, I would sit and look at these things and use these sort of scoring functions in drug discovery projects with our other drug discovery scientists and chemists. And so this has, of course, had huge impact and value for industry. And I think the third aspect of this, that this is no longer an academic endeavor. These programs and these environments are made by small companies that sell them to other companies for money. So it's creating a market. And it's generating a lot of value back to society. And I think, which is important for us as a research community, we need to be able to tell these sort of stories back to the funders for them and not the least the treasuries to understand that actually there is value from the science. And it spills over not only into new interesting science, but actually de facto it helps us make better medicines. And there is a very clear correlation between open, accessible data and better medicines. It just takes, in this case, 25 years. And so, so it's a difficult to quantify correlation, but there is a value there. A more direct correlation is, of course, from these databases, is that you can use them to identify things. And so this is an example from a patent. It's a Dutch company who's sought to improve drought resistance in plants. And so they have found a couple of interesting genes. One of them is ubiquitin protein ligase. And so if you want to patent a plant with that specific gene or a modification to that gene, how do you let the world know what it is? Well, you look it up in the protein database. And so this is what is quoted in the patent. And so suddenly these databases are not only part of a research endeavor, but they really form part of the sort of digital knowledge management infrastructure in a country. They really form part of government legal documents, and they become part of the fabric of what we need to manage in a modern society. And it's just not one isolated example. It is in 2014, there was 117,000 of these citations in different patents. So it's actually used quite a lot. And if you look in some fields, so for instance, in chemicals for drugs, but also for beauty care products, which are interestingly grouped together, then about 20, well, it's floating things with chemicals that you pour on humans, right? Then about 20% of these patents actually used a database to identify a composition. So you wouldn't be able, or it would be much more complicated to write and document this invention for the companies unless this resource would be there. And it's not only drug. If you look at microorganisms, so these are things like washing powder. If you do a washing powder with a new enzyme for taking away fat stains or protein stains, how do you actually claim intellectual property on that enzyme? Well, you actually just point to an enzyme in a database and you say what is different to this reference. And actually about 20% of these patents do exactly that. They point to a reference database and tell the world what they have put into it. So it really is now part of our knowledge management infrastructure. And again, I think this is really important for us as a scientific community to go and tell back to the treasuries and say, well, actually, we've done something which is really valuable. And you need to understand this value because otherwise it will disappear. And so that's part of what we are trying to do collectively to build these sort of value-driven business cases for research funders and the research database owners to be able to preserve it. So with that, I'd like to leave you and thank you for the attention. I hope this was interesting and useful. There are too many people involved in that to thank, so we have about 130 partner institutes. So at our annual meeting is about 250 people coming, so this is actually quite a large network. But thank you very much for your attention.
I do have a prize for, I call it a prize because it's actually an endurance prize for Niklas.
Maybe that should go to the audience then.
Who arrived in Australia last night at about 9 o'clock at night, having flown from London, and this is his third event of today.
Thank you very much.
I would just like to thank Bioplatforms Australia for supporting this event today. And I would, as this is the final Convergence Science Network event for 2015, I'd like to invite the network's Executive Director, Luan Ismail, to say a few words.
Thank you, Andrew. Look, good evening, ladies and gentlemen. I'll be brief. As Andrew mentioned, it's the last event of the year for the Convergence Science Network. It's been a very busy year for us. We've had 12 events and attracted an audience of about 4,800 people for those events. So we've had a new format, as many of you have realised, a new branding, having panel discussions and presentations. We've had 4 of those for you this year, and we'll continue to have that format in the new year. Obviously, the highlight event, I guess, of the year was the Graham Clark Oration with Sir Paul Nurse. Had an audience of about 2,000 people, and most pleasingly, 500 of those were secondary school science students from across Victoria. So that was a really positive outcome, I think, and hopefully will inspire some, scientists, students to do some great things. Obviously, these events would not be possible without the generosity of our partners. Each of them, in their own way, makes a contribution to the sort of innovation and research ecosystem here in Melbourne, but they all come together because they have a passion about public engagement in science. So their generosity makes these events possible. So I'd like you to join me and thank them in the usual way for their commitment. The only thing left for me to do is to wish you a Merry Christmas and a Happy New Year, and we'll look forward to seeing you in 2016. Thank you and good evening.